In the Björkman Lab, we visualize HIV-1 at different stages and on different scales. In one aspect of our work, we use electron microscopy to image tissues from HIV-1 infected humanized mice. These videos show electron tomography of HIV-1 infection in gut-associated lymphoid tissue. Here we can see pools of free virions, as well as the localization of HIV-1 virions within narrow intracellular channels. In a separate aspect of our work, we aim to obtain new insights into HIV-1 entry. The HIV viral membrane is sparsely coated with spike trimers, called envelope, which mediate fusion between the virus and its target, a CD4 T cell. The T cell's surface is arrayed with CD4 molecules, shown here in yellow. Each spike trimer is capable of binding 3 copies of CD4, resulting in large conformational changes in OMV. Zooming in on a single spike trimer, intermediate structures containing 1 or 2 copies of bound CD4 molecules, which must precede full CD4 saturation, had not yet been visualized in high resolution. Using trimers designed to bind to only 1 or 2 copies of CD4, we visualized a single copy of CD4 bound to a closed trimer, and 2 copies of CD4 bound to a partially open trimer. Returning to our schematic of HIV-1 entry, we are also interested in how immune molecules, called antibodies, bind to the spike trimer and block its function. We are interested in antibodies that arise naturally during HIV-1 infection, as well as those that are elicited in response to engineered vaccines. Shown in red are 3 copies of an antibody fragment bound to a spike trimer. Structural biology of HIV-1 often leads us to unexpected findings, and it is our job to better understand the implications of these surprises. The ability to visualize HIV-1 during an infection, coupled with the elucidation of high-resolution structures governing viral fusion, not only enhances our understanding of fundamental biology, but also holds significant promise for the development of novel strategies to treat or prevent HIV-1 infection.